Hi, everyone. In today's lecture, we're going to tackle the topic of the origin of life on the planet Earth. To do that, we have to start out with a simple definition, or maybe not so, of what it means to be alive. Now, in different classes, we might expect different answers, what it means to be alive. But in a science class, we're looking for the biological definition of living organisms. So how do we distinguish between rocks and minerals and those things that are considered to be alive? We winnow it down to at least 6 different components that must be present for an organism to be considered alive. They're usually composed of cells. All living organisms that we know of are composed of cells. They have an internal metabolism. They take in energy and they burn it to operate their cells. They have what is called homeostasis. That is, that they have stable internal conditions that are different than the environment around them. So the cell isolates them from the environment around them. They have the ability to grow through time. They reproduce using heredity, and we'll talk about what types of heredity there are. And they respond to a stimulus. So all living organisms, when the environment changes, they provide some sort of response to that stimulus. So if an organism matches or meets all of those different components, then it is considered to be alive. There are some gray areas, things like viruses, which are abundant in the news today. Discussion is abundant in the news today. Viruses don't meet all of these. They don't have a true metabolism. They use the energy of the cell that they invade. They don't reproduce on their own. A virus by itself simply can't reproduce. But once it's in your body, it attaches itself to a cell and hijacks that cell's reproductive capacity, makes copies of the virus, which then explodes the cell and releases many, many copies into the organism, and then repeat. And you end up with a viral infection. So even though viruses feel very alive when they're in you, they don't typically meet the basic requirements for being considered alive. So that would be a gray area for discussion. Everything that we're going to be looking at are fossils that are derived from once living organisms. So they show conditions of true life and evidence of true life. As far as the origin of life is concerned, it occurred relatively early after the formation of the Earth. So we're going to zoom back down into the Precambrian, the oldest era on our geologic timescale that we're going to follow throughout the semester. The Earth itself, as is discussed in another lecture, is about 4.55 billion years old, or 4,550 million years old. Relatively quickly after the formation of the Earth, we have the formation of life. So we see the Earth form 4.5 billion years ago, and then within the first 300 million years or so, we have the formation of liquid water and oceans. Oh, the first question we should ask ourselves is that why Earth? None of the other planets in our solar system have life. We're investigating Mars and expending a significant amount of our scientific endeavors looking at Mars to see if life once could have lived there or did indeed evolve there in similar ways to what happened on Earth. But Earth itself is the only planet that we know of with direct observation that contains life. So why Earth? It's not a coincidence. Earth is in the prime zone for the chemistry of life to take place. If you look at our galaxy, there's a galactic habitable zone. It's highlighted in the image. There's a little green area. If you're closer to the center of the galaxy than that, there's too much energy. If you're farther away, there's not enough. So in that green zone, it's what's known as the Goldilocks zone. It's just right, not too hot, not too cold, so to speak, not too much radiation, not too little. So that's our Goldilocks zone within the galaxy. If we zoom down into our solar system, there's a similar Goldilocks zone within our solar system. It's just outside the orbit of Mars and just inside the orbit of the Earth. So Venus, for example, is not within our solar system's habitable zone. The planet itself is too close to the sun. So why Earth? Let's look at some failed planets that could have the potential for life but don't have the right conditions. Venus, for example, has because it's too close to the sun, has too high a surface temperature. And therefore what happens is, as Venus is operating, as plate tectonics is taking place on Venus, there are lots of volcanic eruptions that dump gases into the atmosphere. And those gases then aren't allowed to back react with rocks because they don't have liquid water. So what the Earth has that Venus doesn't have is standing oceans of liquid water that allow things like carbon dioxide to dissolve into and then precipitate out in solid form as rocks. Instead, in Venus, CO2 gets trapped in the atmosphere and temperatures escalate and are very, very high, too high for life to have formed. Mars is within the habitable zone. So if it was a little bit larger, it's possible that Mars would have life, some other type of life than what we know on Earth. Instead, what happened is, because Mars is small, its plate tectonic cycle died early. So volcanism stopped, and there was no longer a dumping of carbon dioxide into the atmosphere. And the atmosphere itself that was there was stripped away and was not replaced, because when plate tectonics stops, you lose your protective magnetic field that helps to deflect the solar wind. It would strip the atmosphere away from the planet. Earth has an operating plate tectonic system, which allows gases to be dumped into the atmosphere. It's cool enough that the atmospheric gases can be dissolved and reprecipitated as rock in the oceans. And as there is an internal flow of liquid iron around the solid iron core, that generates a magnetic field that protects our atmosphere and allows conditions to be ideal for life to form. So Earth is the only planet within our solar system that is primed for life. It's not a coincidence that life evolved here. It's possible that there are micro conditions on, say, the moons of Jupiter or places in other galaxies that allow life to evolve. These are just the ideal settings that make it most likely for life to have evolved. So we zoomed back to the Earth when this began to occur around 4 billion years ago, 500 million years after the formation of the planet. If I were actually in this environment, I'd take my first breath and then die, because the atmosphere contains carbon dioxide, nitrogen, and water vapor. You'll notice the lack of oxygen in that case. So early Earth would have been very different than what we see today. There's an evolution of oxygen within the atmosphere that life takes advantage of. But the earliest life to evolve on Earth must have been life that did not need oxygen, because oxygen wasn't present there. There are some proposals for where we think life may have originated. As far as the list is concerned, there are geothermal pools, places where there's lots of energy and water. So on your screen is the Grand Prismatic Pool in Yellowstone. You see bacteria growing at different temperatures within that water. There are hydrothermal vent proposals. Those are deep within the ocean, a couple miles down, where there's an input of energy from magma near the surface that generates the right conditions and chemicals needed for life to have evolved. It's also protected from the harsh UV rays. Early Earth would not have had an ozone because there's no oxygen in the atmosphere, so there would have been no more UV radiation at the Earth's surface. Warm, shallow seas, so in the top layer of water, about 10 meters or so below the surface, life could have evolved in the early seas that formed on the planet Earth. And then my least favorite, in the upper right-hand corner of your screen, is panspermia, which is the idea that life evolved somewhere else and was transported to the Earth already alive, and then took off. I don't like this one because it doesn't help answer the question of how did life evolve, because if it came from somewhere else, we have no idea where, and there's no way to figure it out. Whereas we have lots of evidence for how life could have evolved on Earth. So it isn't necessary to use an extraterrestrial source of life and then bring it into the planet. There are certainly sources of organic molecules and oxygen and water that are being brought into the Earth by extraterrestrial things like comets that are crashing in very early in Earth's history. But we don't see evidence that an already living organism was transported, a cell was transported to Earth and took seed. So I'll cross that one off the list as one of my least favorite. It isn't as logically arguable as the other three are. In order to sort of gain a better understanding of how life originated on the planet Earth, scientists through the years have tackled major misconceptions about how life may have evolved. For example, Louis Pasteur, who was famously came up with the process of pasteurization, and other chemical advances in science. He did an experiment to try to disprove the concept of spontaneous generation. Spontaneous generation was an idea floating around in the 1700s and 1800s that life was continually appearing from nothingness. And so that when you saw maggots crawl out of rotting meat, that they had spontaneously generated within that meat, conditions were right, and then they grow and appear out of it. Same thing with spoiled broths, things like that. The organisms in there must be appearing from nothingness. So Louis Pasteur disproved that with a relatively simple and elegant experiment. He took some broth, he boiled it in a flask, and he sterilized it. Then he left it open and it spoiled. Microorganisms grew within the broth and spoiled it. Then he took another flask that was vacuum sealed and he boiled it, sealed it, and no organisms ever appeared. So it was a way of preserving the organic matter within the broth and not allowing other organisms to invade it. So that stayed unspoiled. And then he took a gooseneck necked flask, which is the one with the long S-shaped neck on it that you see there. And he boiled that broth, sterilized, and it stayed sterile as long as the gooseneck was present. Gooseneck had a U-shaped turn in it that doesn't allow airborne microorganisms back into the. Even though the neck is open to the atmosphere, it doesn't see any spoilage. Once he broke the neck off, it immediately spoiled the broth. It gained microorganisms that were in the atmosphere as he had shown. So they weren't spontaneously generating there. They were pre-existing organisms that were spoiling and getting into the broth. So life arises on the planet once and then progresses from there, but there is no reappearance of life spontaneously from nothingness occurring anymore. Other experiments to try to generate an idea for how life evolved on the planet Earth progressed. In the 1950s, an individual named Stanley Miller performed some experiments to see how life on the early Earth could have evolved. He set up a model experiment for what we thought Earth would have been like back then. So his composition of the atmosphere was wrong, but that's okay. The concepts that he was deriving are still pretty interesting. He set up a flask that had gases in it, and he had an electrical spark because he thought lightning would have been relatively common, and we still think that's the case. What atmosphere there was would have been disturbed much more significantly than it is today. He had water vapor that was coming from a separate flask that he called the ocean, and he was heating that as the old ancient Earth would have been. So he was bubbling water through and into the atmosphere and then condensing it using a cooling coil and trapping that water to see what kinds of molecules might form inorganically from the atmosphere through these processes. What Miller was able to derive was a number of different amino acids within his trapped water. So he showed that you could form organic molecules, the basis for life, through inorganic processes. What we now know today is that those organic molecules are common even in celestial bodies. On the right-hand side of your screen is a chunk of a meteorite called the Murchison meteorite that was collected in Australia. And it was broken down, chemically analyzed, and what you see are little globules that are effectively liposome layers or cell layers. There's nothing inside them, so they aren't living cells, but they're the same chemistry as what your cells are made of today and what all living cells are like, lipid bilayers. So we can get these cellular materials forming in space. They certainly could have formed on early Earth. So that takes care of the cell basis of life. Cells form, these chemical layers form naturally in inorganic components, space in early Earth, and solutions within Stanley Miller's vials. So this is one check mark off. We can form a cell. Slightly more complex than that is the internal mechanism that allows heredity to take place. All cells that we have ever analyzed on the planet use the same genetic code, DNA, deoxyribonucleic acid, and there's the same 4 nitrogenous bases that allow DNA to retain information. Similarly and related, there's RNA associated within our DNA heredity. And you're going to watch a short video that'll explain why RNA may have been the very first genetic material, and then DNA came second. And in that video, you'll see how inorganic chemistry is related to biology, that biology is just complex chemistry associated with life, and that the two sciences are not disconnected. They meet through a field called biochemistry, where we see the formation in inorganic chemistry of organic molecules then take off into organic chemistry and the chemistry of life. What we know is that the very first cells to evolve were probably archaea. The earliest organisms on Earth are these prokaryotic, very simple cells that have different modes of life, obtain energy through very bizarre methods of converting CO2 and hydrogen to methane, for example, or by the reduction of sulfur components. From there, we evolved more complex cells like the eubacteria, true bacterial cells, and we see evidence of those around 3.5 billion years ago. I'll show you examples of them. The very first cells probably had some pretty simple metabolic pathways, absorb nutrients directly from their environment. They may have gone through the process of fermentation, one of my favorite metabolic pathways. What we know is that they were prokaryotic. So on the left-hand side of your screen is a prokaryotic cell. You see them appear in the fossil record around 3.5 billion years ago. They don't have a nucleus, so their genetic material is in one continuous looped string on the inside of the cell, usually bundled up and wadded up like a rubber band that's been rolled around in your hands. And they reproduce their cells by what's called binary fission. It's a cloning process. There are mutations that take place. So bacterial cells still evolve, but it's much more slowly. The other types of cells, the cells that make up our bodies and other multicellular organisms, are called eukaryotic cells. We don't see those until around 2 billion years ago, and they don't become really abundant as multicellular organisms until around 0.7 to 1 billion years ago. What they have is a nucleus and other organelles. So they have systems within their cell that act as energy resources. So produce energy for the cell, dispose waste for the cell, that store the genetic material for the cell. So they have little components within the cell that do different jobs, unlike the prokaryotic cells. So these are able to reproduce both asexually and sexually, and you're going to have a lecture on metazoans that will go into the eukaryotic cell in more detail. But for today's lecture, we're going to stick to the prokaryotes. They seem to be the very first life on the planet Earth, and we have fossil evidence of these cells. It's amazing that 3.5 billion years worth of history has taken place, and yet you can still have rocks that preserve the very structure of microscopic organisms like bacterial cells. William Schaff in 1993 published a paper that showed the very first preserved and recovered single-celled life. These living organisms are fossils of what is a still living branch of bacteria known as cyanobacteria, present in the green little circles that are all linked together in chains there in the middle of your screen. Those are cyanobacterial cells. We see evidence in the morphology of certain fossils 3.5 billion years ago that show the same structures as these cyanobacteria. There may be even older evidence for life. There was a paper published a few years ago that showed 3.77 billion year old archaic bacteria, those little red tubes that are projecting out on your screen are very similar to the same things we would see in types of iron-reducing bacteria that live in the deep water hydrothermal environments. So those may be fossilized cells of archaic bacteria that are even older than Schaff's cyanobacteria. That's the pathway that we think occurred, archaea to eubacteria. So that would reassuring. There needs to be more evidence and validation of that site to make sure they really are cells. There's also a site in Isua, Greenland that's 3.8 billion years old that has sedimentary structures that look a lot like the oldest types of fossils that we see, that don't have cellular fossils but have structures within the rocks that are left by single-celled organisms like bacteria. We call those stromatolites, and this is a much more elaborate example of a stromatolite. They're fossilized bacterial mats. So bacteria grow in these structures, and we can see them in the oceans today. And as those structures form, we can interpret them as having been built by bacteria. In the modern world, you can go to places like Shark Bay, and you can see these stromatolites. Each of those little mounds is a lump of bacterial colony that's growing continuously. So you cut one open, look at it through the microscope, you'd see it was chock full of cyanobacteria on the outer layers. The inner layers are predominantly just layers of sediment. The bacteria are sticky, sediment washes into the bay, they get stuck to the cyanobacteria, and they form layer after layer as the colony expands out on top of the old layers of sediment. What's cool about stromatolites and cyanobacteria as some of the oldest fossils is that they are the reason that life evolves the way that it does. They're one of the very first organisms to significantly change the composition of the atmosphere, and in this case, at a detriment to themselves. So maybe not that different from our story. But what happens is, as the stromatolites are forming, as these cyanobacteria are photosynthesizing, they dump oxygen into the atmosphere. It's carbon dioxide plus water equals, with a little bit of sunlight, equals sugar plus oxygen. That's the effective equation for photosynthesis. These cyanobacteria, as they're photosynthesizing, are producing oxygen as a waste byproduct. Now, oxygen didn't build up right away. Cyanobacteria appeared 3.5 billion years ago. We don't start to see significant oxygen in the atmosphere until 2.5 to 2 billion years ago. So there's a lag between the production of oxygen and the buildup of oxygen in the atmosphere. And what's going on is these organisms, so as we see, if you look at where the oldest stromatolites are versus where the blue line goes up, oxygen begins to build up about 2.5 billion years ago and then climbs to present day levels somewhere on the order of 500 million years ago. That lag between the evolution of cyanobacteria and the production of oxygen is the result of a process called BIF formation. Banded iron formations are what is sucking up the oxygen that's first produced by the cyanobacteria. Oxygen gets dumped into the water. And the ocean back then would have been very different from what it is today. There would have been free iron. Iron would have been present mixed within the oceans. As oxygen hits that iron, it rusts it, reacts, and creates things like hematite. That's the red mineral that you see in the rocks in this image. That iron-rich environment is then freed of is freed of its iron by oxygen. Oxygen rusts it out, produces these big, thick BIF layers. So if you look at our graph here again, as oxygen increases in the Archean, in around 3 billion years ago, eventually you oxidize all of that iron and the oxygen begins to build up, has nothing else to absorb it, and we raise oxygen levels. That's catastrophic to the other organisms that live in an environment where there isn't any oxygen. Oxygen is a very harsh molecule and toxic to most of these anaerobic bacteria that are living in the oceans. Other organisms evolved to use the oxygen as a metabolite, so they use it to help their metabolism, and we shift the composition of life and change the overall pathway of life by the production of oxygen as a waste byproduct. Now, in our next lecture, we're going to talk about what those multicellular organisms are like and how the eukaryotic cell evolved.